Why is our genetic information encoded in DNA molecules? Why was DNA chosen and not, for example, its older sister molecule, RNA? Is DNA always in the form of the well-known double helix, or can it also form other structures? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Edward Kajnowski, and I work as a geneticist at the Institute of Biophysics of the Czech Academy of Sciences. In previous episodes, we mentioned that the form of the first molecules of life is lost to us far beyond the horizon, and that these could have been RNA molecules. We also said that RNA molecules not only stored genetic information, but also carried it out. In other words, they functioned as catalysts for various reactions. And at a certain stage in the evolution of genetic biomacromolecules, a new medium emerged, serving exclusively for the storage of genetic information. DNA molecules appeared, and they have fulfilled this role to this day. Let's talk about what their advantage is. Why is genetic information stored in DNA molecules? According to some scientists, the DNA molecule was selected during evolution thanks to its unique photochemical properties. It is known that if you irradiate nitrogenous bases with ultraviolet light, they transition into an energetically excited state. The excited molecule then has an excess of energy, which it can either emit, convert into vibrations, or it may undergo a chemical change. Specifically, the nitrogenous bases that are part of DNA, after being irradiated, return very quickly, within picoseconds, that is, 10 to the minus 12th seconds, or even faster, to their original stable state without undergoing any chemical change, meaning the genetic information remains intact. Because during the time life was forming, ultraviolet radiation was much stronger than it is today. Since the ozone layer was missing, the ability of DNA to resist changes caused by radiation was a major advantage. However, RNA molecules also have similar properties. So why did the transition from RNA to DNA occur? The answer to this question can be found in the structural differences between DNA and RNA. Yes, all the rather significant differences in the properties of DNA and RNA are the result of small differences in their chemical composition. The first of these differences is the presence of the nitrogenous base thymine in DNA molecules instead of uracil, which is typical for RNA molecules. The second, and probably more important, difference is the presence of deoxyribose as the sugar component in DNA instead of ribose, which is found in RNA. The point is that deoxyribose is much less reactive compared to ribose, and the sugar phosphate backbone of DNA is not as easily attacked and broken down. This means that DNA molecules are not so easily chopped into pieces by various physical and chemical factors. Higher reactivity was initially advantageous in a nucleic acid molecule functioning as an enzyme, that is, in RNA enzymes or ribozymes. We have already talked about them earlier. On the other hand, in the case of a medium for storing information, which became DNA, it was necessary to ensure its invariability and stability. Therefore, greater chemical resistance was more advantageous. That is also why DNA molecules are much longer than RNA molecules, which made it possible to store much more complex information. Without increasing the capacity of the storage medium, that is, without lengthening the DNA or enlarging the genome, life could not have reached the level of complex multicellular organisms as we know them today. The choice also fell on DNA thanks to its ability to be copied more accurately compared to RNA molecules. It has been proven that the chemical differences between RNA and DNA make the elongation of DNA molecules much more precise when a DNA molecule is used as a template. If DNA is synthesized from RNA molecules or RNA is copied from its own template, the incorporation of nucleotides into the elongating nucleic acid strand is much more error-prone than when a DNA molecule serves as the template. Errors also occur during transcription, that is, the creation of RNA from a DNA template. The accuracy of both replication and transcription was initially low, and it only improved after the discovery, in quotation marks, of protein catalysts, including various repair mechanisms that oversee the precision of molecular processes. However, a certain level of transcriptional error can actually serve as a source of desirable phenotypic variability, meaning different ways of expressing the same genetic information that ultimately lead to slightly different body structures. Another advantage of DNA is its ability to form a wide variety of spatial arrangements. Sometimes the DNA double helix is coiled like a right-handed staircase, known as bDNA or aDNA, and at other times it forms a left-handed staircase known as zDNA. Sometimes it doesn't even settle for just 2 strands, and a 3-stranded DNA known as a triplex can form, or even a 4-stranded structure called a quadruplex or tetraplex. Different forms of DNA can function as molecular switches for a variety of biological processes. Some researchers in the field of nanotechnology who operate in the world of truly tiny dimensions, a nanometer is 1/1000th of a meter, are trying to construct DNA-based nanomachines. It is precisely the switches between different DNA structures, which could be controlled by various small molecules, that are likely to find their use in the future. The DNA molecule is truly a remarkable medium in which several layers of information are superimposed, and each of them has its own code. After all, besides the well-known triplet genetic code, which assigns the sequence of amino acids in proteins to the bases in DNA, there is also the epigenetic code, which, through various tags attached to the DNA, determines whether genes will remain silent or be expressed. But we can also hear about the chromatin code, which determines how DNA is packed into chromosomes, about the DNA bending code, which dictates how the DNA molecule bends in space. We can hear about codes that determine the conformation of DNA, its interactions with proteins, or the sites of DNA recombination. The individual layers of information must be arranged in the linear DNA sequence in such a way that they do not interfere with each other too much, but rather can interact with each other. To conclude this episode of our series, let me just remind you that DNA molecules arose long after the emergence of RNA molecules and the first proteins. It is even assumed that DNA only came into play after the emergence of the first cells, the dating of which remains one of the greatest mysteries in biology. DNA is simply a latecomer, and perhaps that's why, unlike RNA, it has only one main task, one role, to serve as the cell's repository of genetic information. So that's all for today. In the next episode, we'll talk about whether life could, in principle, be based on elements other than carbon, such as silicon. I'm really looking forward to seeing you. If you enjoy my talks, I'd be grateful for a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe, which will help these videos reach more people. I really appreciate your support. Thank you and bye-bye.